There are 4 levels of protein structure, and it's important to know what each level of structure means and what interactions control the shape of the protein at that level. The primary level is simply the sequence of the amino acids. Remember that they have a condensation reaction that forms this peptide bond or an amide functional group. And the order of these amino acids is defined by the RNA and DNA that is encoding these particular proteins. So know that primary level is just the sequence, and know that you read it from the N terminus over to the C terminus. And so that's how amino acids are read in sequence. The secondary structure is the interactions of essentially the backbone. Notice that you can have either alpha helices or beta pleated sheets, and you can have these at different regions within the same protein. It's basically the most stable way for these functional groups to orient themselves most comfortably. Both the alpha helix and the beta pleated sheet are created by hydrogen bonding between the hydrogen here as part of your amino group and the carbonyl oxygen on the other side of the amino acid chain. It can either be an alpha helix, which winds in a helical shape, or it can be a beta pleated sheet. But both of these are defined by the hydrogen bonding between that amino hydrogen and the carbonyl oxygen. Tertiary structure is where it gets very interesting and where you start to see the larger protein structure emerge. A lot of proteins never go beyond their tertiary structure, but the factors that influence the tertiary structure are the hydrophobic and hydrophilic interactions, how these polar and nonpolar groups interact with water. There's hydrogen bonding that's involved, there's ionic bonding, the cysteine-cysteine disulfide bridge that helps anchor the structure, and all of these establish what is called the conformation. Conformation is the 3-dimensional folded structure of the protein. The really interesting thing to realize is that if you find a way to denature a protein, either by adding heat or by adding some agents that cause the protein to flatten out into a long strand again, usually it will reform its conformation once the denaturing agent is removed. And that just shows you how much these electrostatic interactions are influential for determining the shape of your protein. So when you think of tertiary level structure, there are a lot of factors that are involved. Hydrogen bonding, van der Waals forces, ionic interactions, and everything, all of the side chains interactions with water. All of those things play into the complex 3D conformation of the protein. The last level, the quaternary structure, is something when you have a protein that involves multiple subunits. And the prominent example that you'll see of this on the MCAT is going to be hemoglobin. They may describe other proteins with quaternary structure, but the one that you'll be responsible for is hemoglobin. You'll just have to realize that it has 4 distinct subunits that all fit together. A lot of proteins do just fine without a quaternary structure, but if there is one, then it's a complex protein that involves a lot of pieces coming together. So recognize all of these different levels of protein structure and the bonding and interactions that allow all of these to exist. Primary is defined purely by the sequence of amino acids, and that's pretty much set in stone by the DNA. Secondary is these hydrogen bonds that allows the helix and the pleated sheet to form. Tertiary is the very complex set of interactions, and quaternary is when you have multiple subunits. Later on, as you advance and start to discuss more about proteins on the MCAT, you'll find a lot of questions ask you about disrupting some of these interactions, and it'll be your job to figure out what level is being interrupted and how that influences the function of the protein.